Hello students, today we will learn what is ASO-RDB, the full form being allele specific oligonucleotide reverse dot blot hybridization. This is a very widely used technique in detecting various point mutation related to various genetic diagnosis. Okay, so let us first understand these two terms so that it will be very easier for you to understand them together. So first we should understand ASO, allele specific oligonucleotide, as the name suggests. So this is the definition is actually a short fragment of synthetic DNA that is complementary to the sequence of a variable target DNA. Very important. So the synthetic DNA that we need to design, okay, that should be complementary to the sequence of a variable target DNA. Means we should know the target DNA and we should design the synthetic DNA so that it is exactly complementary to that one. Okay, so thereafter you can simply, at this point of time, you can simply pause this video and read along, because we don't need to read everything. It will be very clear as we go along as I describe you diagrammatically. So consider these are the two alleles. One is normal and one is mutated. So we consider this one as the normal type. Normal in genetics is also known as wild type. And here this is the mutant type, where this single nucleotide A has mutated to G, and likewise its complementary T has been mutated to C. So this is the site of the mutation. All right, so how can we detect it with the help of this method, ASO-RDB? We are designing probes, as I told you, specific probes, that is sequence of a synthetic DNA that is complementary to the variable target DNA. So we need to design probes that are exactly complementary to the mutant sequence, and we also design a probe that is complementary to the wild sequence. Okay, so we now have two probes. No problem in understanding it. So what happens? We expose the sequences to these probes. Okay, very easy. In this case, it has been shown with the probe M. Suppose mutant probe. So what will happen? Mutant probe will bind perfectly to the mutant allele, as it is absolutely complementary. However, it will not be perfectly matching with the wild type allele. There will be a single base mismatch in binding. However, it will still bind, but the concern is, as we change the condition, these conditions can be changed in temperature or pH, whatever it is. We can design the experiment based on that. Then this DNA heteroduplexes that are perfectly complementary for in case of the mutant allele, in this case, will still remain intact. For example, here we are showing the rise in temperature near the melting temperature. So if in this case we are reading the experiments or reading the observations with probe M exposure, the duplexes are still intact. However, in case of the probe W, the duplex, due to mismatch, they have dissociated. All right, the same could have been if we are giving probe W, that is wild type probe. The match would have been here and this duplex would have been stable. However, this would have been dissociated. So this is the concept of allele specific oligonucleotides. Okay, so we are first designing this allele specific oligonucleotide. Now let us understand reverse dot blot hybridization. First of all, we need to understand what is dot blot. Okay, then how is it reverse. So you can see the first line, we are starting with a negative word, unlike. So unlike traditional dot blot, allele specific oligonucleotide probes are immobilized on a membrane and patient DNA is hybridized to detect the mutation. Again, over here I just urge you to pause this video. After you have understood the whole thing, you can again rewind and come back to the slide and then read it through and then prepare your own notes. Okay, so let us go forward and see the diagram. So in the left side, we are showing a traditional dot blot. Mind it, in any case of blot, here if you are detecting a DNA, what do? We expose or we immobilize a DNA on a or DNA of interest on a nylon membrane. Okay, and then we add probe on top of it. Okay, so those probes will then hybridize to the DNA of interest. So those probes may be labeled probes. Here see biotin labeled primers, and then the target DNA will be amplified with primers, and the probes will be labeled, and then it can be detected by a signal. Okay, just understand the concept over here. Here we are discussing reverse dot blot hybridization. I was just discussing dot blot. So here what we are doing, we can, so here it is showing a radioactive probe that can be detected by autoradiography. Had it been a biotin labeled probe, it could have been detected by streptavidin HRP. So whatever it is, we are exposing the DNA on the membrane, and we are exposing it, treating it with the probes. In case of reverse dot blot hybridization, what is happening, we are immobilizing the probes on the membrane, and we are adding the DNA of interest later. So it is simply the opposite. The process is just the opposite. In traditional blot, we are first adding DNA and then we are adding probes. In case of reverse dot blot, we are first adding probes, then we are adding the DNA. Okay, what DNA? Patient's DNA that has been amplified using PCR with labeled primers, right? We cannot add simple DNA because DNA is very, patient DNA will be very less. So that is the importance of PCR. We can make 1,000,000 copy to increase the detection level sensitivity. So what happens in case of ASO-RDB? We can take a membrane, and suppose in this case we have we have given 5 dots of a wild type probe on the top, that is normal. Okay, ASO, and we have given a dot, or we have immobilized 5 dots of a mutant probe on the membrane. Why we have given 5? Because it is the art of multiplexing. We can do simultaneous analysis of 5 patients in this case, or multiple patients, or in duplicate or triplicate of a single sample to cross verify. So what is happening, consider here are, now we are adding the DNA which has been amplified by PCR with labeled primer, biotinylated patient DNA. So you can see, even without me telling you, you can easily understand that in case of number 4, what has happened, the DNA has bound only to the mutant probe, and not to the wild type probe. It means there is no mutation. Okay, in case of number 3, what has happened, the DNA has only bound to the mutant and not to the normal, means there is a mutation, not only that, it is homozygous. Whereas in case of patient, or case 1, 2 and 5, we can see that the DNA has bound to the both the probes. Okay, it means there is a heterozygous mutation. So this is actually a case of cystic fibrosis. Okay, so cystic fibrosis that can be diagnosed by ASO-RDB. Multiple hemoglobinopathies can be done, detected by ASO-RDB. Okay, so these are all PCR based method. So this is all about the ASO-RDB. Now we need to know the advantages and limitation for exam. It is very easy to visualize advantages being simultaneous detection of multiple mutation. We can detect multiple probes because we know what are the variable target DNA sequence, and to them, complementary to them, we can design the allele specific oligonucleotide. And since the sequences are known, it is highly specific and sensitive for known mutation. Very, very, very important, known mutation. And you can see we have added 5 dots to a membrane, right? And then we are adding the DNA. You can automate the process. We can add hundreds and hundreds of dots, and we can use robotic arm to simultaneously apply multiple samples, right? So it is automation friendly, making it ideal for large scale screening. Okay, limitations, definitely it has its limitation. One very important thing is it cannot detect novel or unknown mutations. It cannot. So it is defined to, limited to predefined mutation panels. If examiner asks you, so what will you do in case of novel or unknown mutation? Answers of course will be gene sequencing, either Sanger sequencing or pyro sequencing, that is next generation sequencing, whatever it is. So that is not the topic of today's discussion. Definitely I will be covering many more such mutation diagnosis videos in my channel. So that's it for today. So I hope now you have understood ASO-RDB. Please ask in the comment section if there is any query, and please like the video if it has helped you. If it has helped you to understand the concept, please share with it your friends, and please subscribe to the channel if you are not subscribed. I'll see you soon with another video. Till then, bye and take care.